Hello, everyone. My name is Gerard Gatch, and I'll be the moderator for today's webinar on the topic of enrich, an automated process of enriching side fractions or impurities. Okay, today's presentation will cover these topics: enrich, the enrich technology, how it works, a few case studies, and then we'll go on to a summary and your questions. Today's presenter is Thomas Müller-Spath, who holds the position of Chief Technology Officer at YMC Chromacon based in Zurich, Switzerland. Thomas is an inventor of more than 10 patents and has authored and co-authored more than 30 scientific articles and book chapters on continuous chromatography for biopharmaceuticals. Thomas frequently presents at international conferences as speaker and also co-chairs workshops on continuous chromatography. Please welcome Thomas Müller-Spath. Thomas?
So hello, welcome also from my side. To this presentation about enrich, about the automated purification of impurities and side compounds. So why are we doing this? What are we using this technology for? So the background is we have in many cases of proteins, but also peptides or other compounds produced by chemical synthesis, a variety of side compounds that accompany the product as it is produced. So our products are heterogeneous mixtures generally. So the one aim of the continuous of the downstream process is to, well, first of all, reduce heterogeneity if possible, but in many cases also to maintain a certain impurity profile. Now, once we maintain a certain impurity profile, it means that we have to characterize our side compounds that are in there. So this is a regulatory requirement. And as you can see here, these are profiles, example profiles from proteins showing a glycosylation pattern or a charge isoform pattern for monoclonal antibodies as shown on the right-hand side. So this actually shows an experimental chromatogram obtained from analytical cation exchange chromatography, where side compounds, antibody isoforms are very prominent. And one typical job to do in our life as downstreams specialists is to maintain a certain milligram quantity of certain isoforms within a certain period of time. So preferably as fast as possible. Let's say we need to produce a couple of milligrams of this peak here. So how can we achieve this? By loading more on the analytical column? Probably not, because then we will lose resolution as the peaks are just growing and overlapping here. We don't get our compound pure enough. So in real life, the workflow looks like this. So we have a certain starting material. We do some preparative HPLC or FPLC, whatever, and we do that at a low load. We fractionate the peak. We analyze the peak for the isoforms. So we do a fine fractionation. We collect the isoforms in certain fractions, so the isoforms that we want, and then we pool those fractions. And then we start over and over again. We do that over and over again until we have produced sufficient quantities of our isoforms that we want. So, and this can take many cycles, and it can take also a couple of days. So we hope then also at the end that what we have produced on day one has still not degraded, let's say oxidized, deamidated, or whatever, degraded in some other form, so that we still have can mix it and still have representative material with compared with runs that are produced have been run at a later stage, and material that has been produced on later days. Okay, so we are suggesting here a process that's called enrich. This process runs multiple cycles automatically to enrich side compounds. And the way it works is we first identify the location of our impurity, then we run the process to enrich the impurity inside the process, then we do analytical HPLC or UPLC to characterize the profile that we have generated in the process. And I will tell you how it works in a few minutes. So the advantage of this process is that we avoid tedious collection of fractions from analytical runs. So we have much fewer runs, much fewer sample. We deplete the undesired main compounds, and we save a lot of time by avoiding this repetitive work that we would do usually as shown on the slide before. And also because we are so fast, we are preserving integrity of the desired components through fast isolation. Okay, so the enrich process has 3 phases. First, an accumulation phase where we grow a certain compound of interest, so a certain region of the chromatogram is selected for enrichment, and then it's grown inside the process. So the concentration of the product is increased, and then this accumulation phase is followed by a separation phase where the concentration of this compound of interest is kept constant, and the other compounds that we do not want are decreased in concentration. And then there's a final elution phase where we run a gradient, a shallow gradient, and do a fine fractionation. And those are the fractions here that we obtain in the end that we actually analyze to determine if we have received our compounds of interest with sufficient purity. Okay, so 2 of these phases are actually cyclic, so that's shown here. Just schematic looks kind of complex, but we'll have an animation on the next slide. What I want to show here is this: we have a first cyclic phase where we inject, every cycle we inject feed, so we inject new material that contains our side compounds that we want to isolate. So here we have feed injections, and we run that a couple of cycles. Then in the second phase, the separation phase, we deplete the unwanted compounds. We do not inject new feed. And then in the final gradient elution, also we do not inject feed. We just do a single step for eluting the 2 columns in series, and then we do a fine fractionation. Okay, so this, as you've seen, this is a cyclic process. We need some equipment for that that can run this process fully automated, and that has also an operating software that can run this kind of process. And this equipment is shown here. It's called the Contichrom Cube equipment. It goes up to 36 mils per minute, and it has UV detection capabilities and goes up to 100 bar. This instrument is used for continuous chromatography, and the enrich application is an application that you can run in addition to the continuous preparative processes like capture SMP and MCSGP that have been presented in the last webinars. In addition, this system, of course, can also run single column chromatography or 2 chromatography steps in a row, like protein A and size exclusion with 2 different columns, fully automated. This case study that I'm presenting here, in this case study, we used 10 micron small particle Smart Sep S10 resin from YMC. This is for purification of isoforms from monoclonal antibodies, and that was packed in ChronLab Eco Plus glass columns, which have a high pressure rating of up to 80 bar. So now I'll show you how the process actually works using this nice animation. So here we have our 2 columns packed with the same material, the small particle cation exchange. And you can see this is a chromatogram, schematic chromatogram of the mixture where that contains our side compounds of interest, which are here shown in red color. So let's say this is the, so to say, the product, the side compound that we want to obtain. And we also have overlapping portions of the product, of this product, with side compounds that we do not want, and we have these pure side compounds which we consider waste in the front and in the end. So we have waste, overlap, product, overlap, and waste. And this is present as a mixture in the starting material, which could be, for example, in the case of monoclonal antibodies, a protein A eluate, a neutralized protein A eluate. So we load this on the first column, and we reject or elute the waste and the overlap. Now we have our compounds of interest, which are at the exit of this column now. Instead of eluting also these compounds, we internally recycle them by an interconnection from the first column into the second column, and we inline dilute this stream on its way to the second column, so that these isoforms here contained in this portion reabsorb at the column entry. So they basically stick at the column entry of this second column now. And so we've kept them inside the process. Now, we see at on the first column, we only have compounds which are not of interest remaining, so we elute those and we re-equilibrate, or the first column. We can also clean it first and then re-equilibrate it. So we make it ready for the next injection here, which actually takes place now in one of the next steps. So first, let's focus on the second column here. We put new feed, we inject new feed on the second column here. And you see now the concentration, and we run a linear gradient. So the concentration of this compound of interest has now increased. You can see it's a little bit brighter now here in terms of color. Again, we elute the waste and the overlap. And now, again, comes at the outlet of this column the portion of the chromatogram that contains the compounds of interest. We want to keep this in the process. So again, we interconnect the two columns and we continue the gradient on this second column here, so that the compounds of interest elute and they are injected onto the first column, which has been previously re-equilibrated. Again, inline dilution is present to make sure that this portion, which has been desorbed from the second column, actually adsorbs on the first column. Okay, so now, again, we have kept the compounds of interest inside the process, and now in this column we only have compounds remaining which are not of interest, so we clean that column and we re-equilibrate the column, and now we inject new feed on the first column, and you will see now the concentration again increases. We'll get a nice bright red here of the compounds of interest, and you can see now that we are essentially at the initial stage. We have just amplified a certain region of the chromatogram. We could go on forever like this until all the starting material has been consumed, or until we have produced enough of our side compound here. So once we have produced enough, we want to get it out of the column, so we can do a final elution to elute the enriched product here. So that's basically how it works. The animation is a bit simplified, but basically that's how the process works. Now, this process applied to monoclonal antibodies is shown on the following slide. So let's say our aim here was, again, to produce 1 milligram of each of those isoforms. We have acidic isoforms, we have basic isoforms here, and these are present in relatively, in different amounts, the least abundant one being present with 0.5%. So it's quite a challenging task to get that one. That's the one here in the front. So now using a wizard tool, which is included in the software package that comes with the Contichrom instrument, we can actually produce the method, or generate the method for running the enrich process in a very short period of time. So we start from a single column batch chromatogram, and in this chromatogram we select by drag and drop a region of the chromatogram that we want to amplify. So let's say here in the software tool, this is our chromatogram that we have run on the system, and now using the software tool we load the chromatogram and we select a certain region that we want to amplify. So it's purely by drag and drop. So then in the next step we set a desired washing and cleaning protocol, how many column volumes we want to wash, how many column volumes we want to clean and re-equilibrate, which pump, which buffer, all of that is set here. We can take that directly from the single column method that we have run here. And then we do some final settings in the end where we set the final gradient dilution parameters and the fraction size. And actually, before doing that, we set the number of cycles for accumulation. So again, this is the chromatogram now here shown in the zoom, where we select a region for enrichment. So let's say we want to go for the acidic isoforms, so we select the front region of the chromatogram. Now, when running the process after having generated the methods, we see now the following chromatograms. These are typical chromatograms that we get from Enrich. So we see two UV signals from column 1 and from column 2. Each column is equipped with a UV sensor, and we see how the profiles grow over time. So these are again acidic isoforms, and you can see how they grow inside the process, whereas the rest of the chromatogram stays constant. Right? So these are the acidic isoforms that now essentially get enriched inside the process. Okay, so after this accumulation phase that you have seen, we run the separation phase. So this is one cycle where we are not feeding any new material. So basically, we are just recycling once the compounds of interest, and we are discarding the main compound. So again, as reference, we have grown this portion of the chromatogram that you can see here in the upper right. So this portion we have grown, and this portion, the main compound, now we deplete completely by not injecting any new feed and just discarding this portion of the chromatogram. So you can see that peak becomes very small. And then we do a final gradient dilution. So this shows you the chromatogram of the final gradient dilution, and it shows you how the front portion of the chromatogram, the acidic isoforms, got enriched. And also shows you that the main compound, which is shown here, has been really diminished to very small levels. So now these fractions then are analyzed, and in one go now we have basically produced many isoforms, and with a fraction analysis we can identify the purest isoforms, and through the increase in concentration, chances are very good also that we have accumulated sufficient quantities of these isoforms so that we can meet our target. So this is a summary now from analytical fraction analysis, analytical HPLC. Shows you the isoforms that we wanted to get, acidic isoforms here. Now we got more than we needed, so we wanted 1 milligram, we got more than 1.5. This one was, the A1 isoform was difficult to get. It's still overlapping a little bit with the A2, so we fell a little bit short of the purity of 90%, but talking to the analytical department, it was still found that this purity was okay enough, so that they could actually proceed with the analysis. So these isoforms then, they can be used for cell-based assays or other testing, mass spec, so on. So the basic isoforms, which are related to lysine variants, they were isolated in a separate Enrich run, and also they were obtained in large amounts and sufficient purity. Here, this slide shows a summary of the individual chromatograms. So here we have isoform A1, isoform A2, and here for comparison also isoforms K1 and K2, so the lysine variants, which were obtained in a separate Enrich run. So this is an example that shows a calculation of the actual time savings that were obtained by using this chromatography technique. So here, in the second example, the aim was to produce a larger amount of isoforms, or 10 milligrams, 2.5% content in the feed was the content of the least abundant isoform. And essentially, you can show that you can decrease significantly the total time required for the isolation by almost, well, by a factor of 10. So, and this is majorly due to the reduction of analytics time. So whereas in the single column method, you have to fractionate every single run and spend hours on your run fractionation. So let's say you have 20 fractions per run, and one analytical run takes 1 hour. You spend 20 hours per run to get the results from a single isolation. However, because you can produce only very small amounts in each run, you need a lot of cycles. So you need to repeat this batch 30 times or 32 times, and then you have 20 fractions per run, corresponding to 20 hours. You need about 600 hours of, well, you have 600 samples and 600 hours of analytical time, whereas with Enrich, you only analyze the final fractionation. You do a finer fractionation, however, you only have 50 samples because it's just one, you just do it once. So that analytical time for the final elution is a bit longer, but still you are down to 50 hours from, or down to 50 samples from 600 samples or 600 hours. And that leads to the massive reduction in overall processing time. And it also, of course, reduces the burden on your analytical department a lot. This technique is not only applicable to antibodies, but also to peptide impurities, where characterization of impurities is also very important, and it's very difficult to get them usually to get them in pure form. So this shows an example of a fibrinopeptide where the aim was to isolate the certain impurity, which is located here in front of the main peak. And this shows you the purest fraction of the single column run, and this in comparison the purest fraction of the Enrich run. And again, the performance of Enrich was very good. The purity was much higher here, more than 80%, whereas the purity of this compound obtained through the batch run was smaller than 20%. In Enrich for peptides that are used in conjunction with the reverse phase, we frequently have this issue that once we load more, the peak moves to the front in the preparative chromatography, and therefore it becomes very difficult to get impurities which are sitting directly in front of the main peak. And this explains, this moving to the front of the main peak explains why we have so much main compound here present in the purest fraction of this target compound. Okay, so with this, I'd like to close the presentation. So Enrich gives you a number of benefits. So automated enrichment, reduced sample handling is certainly one of them. Reduced number of samples, and massive time savings, and associated with that preservation of sample integrity, because the samples are not sitting around waiting for other samples to be purified and blended in, but they can be used very quickly for more important objectives. Also, we've seen in the case of fibrinopeptide that we can get higher side compound impurity, let's say concentration and purity. So again, thank you, and I'll hand it over for Gerard for some concluding remarks before we then move on to the Q&A session.
Thank you very much, Thomas. That was an excellent presentation, very, very informative. This presentation was sponsored by YMC Corporation You might know YMC as a pioneer in chromatography, producing bench and prep scale HPLC and LPLC systems, packing resins, analytical columns since 1980. YMC has manufacturing and offices in over 10 countries. So contact us if you want any more information on feasibility studies or supplying any of these products by YMC. What you may not know is that YMC recently purchased both Chromacon and the former Lewa process technologies, which add significantly to their 40-year history to now lead the industry in the emerging area of multi-column or continuous chromatography. The result of these acquisitions is the corporation's able to provide a seamless platform of continuous chromatography from lab to production scale. Just a reminder that other aspects of this topic are covered in focused webinars, which are aspects of this technology that Thomas just didn't have time to touch on in this webinar, but will provide you with a great deal of insights on some emerging technology around chromatography that you may not be aware of. Okay, we did get some questions. Thank you very much, audience, for the questions. Continue to submit them. I'm going to bring back Thomas for the Q&A, and let me take a look. And the first question we have is, how do I determine how many cycles are needed for enrichment?
Okay, so you determine that basically by looking at the concentration of your compound of interest. So let's say you have, as shown initially, 1% of the feed mixture is your compound of interest, and then you with the starting material concentration, 5 grams per liter, let's say. So you know that 1% of 5 grams per liter is going to be 50 milligrams per liter, right? So, and then with your injection volume that you have, you can estimate how many milligrams or micrograms you are injecting with each injection. So if I have 50 milligrams per liter, I will go down to 50 micrograms per milliliter injection. So I have 50 micrograms per milliliter injection, and so if I want to have 1 gram, again, I need to do 1 milligram, sorry, I need to do 20 injections, 50 micrograms times 20 to get to 1 milligram, right? So essentially, I can, that's the way how I calculate it. I go with the least abundant one, the least abundant isoform that I want, take the content of that, and multiply with the injection volume, and then I get how many cycles I need to run. That assumes 100% yield, so I may need to want to run a couple of extra cycles just to make sure.
Okay, thank you for that. Next question. Do I need analytical resin for the enrich process?
Yeah, so the resin that I showed you is a semi-preparative resin, right? Which means, as opposed to a typical analytical resin that was used in the analytical cation exchange, it does have a decent porosity, so it does have a decent capacity. Some analytical materials have no porosity, just to increase mass transfer. But in this case, we do need to have some, let's say, significant capacity for binding of proteins, or in the case of reverse phase peptides or oligos, for example, also. So we need actually a semi-preparative resin for this process. It does need to have good resolution, though, so small particles is definitely recommended.
Very good, thank you. Next question, is the process scalable?
Yeah, so that's a good question. I mean, it's intended as a tool for analytical or in analytical departments, right? So to produce certain milligram amounts of isoforms, so or other side compounds. So we actually think that it doesn't need to be scalable. So it's certainly not a manufacturing process because it doesn't reach a steady state. So it accumulates product inside all the time, right, or compounds of interest all the time. So we consider it really a tool that is in product development.
Okay. Next question. Do you have a test program where I could send a sample and you would enrich it?
Yes. So this is possible. I mean, we are open to do project of this type. So, and we've done it in the past. So yes, the answer is yes.
So to whoever asked that question, if you would want to follow up on that, you can just send us a note at info@ymcpt.com. That's info@ymcpt.com. And maybe Thomas can carry on that conversation with you one-on-one. Okay, one or two other questions. Is this software that I could load on another manufacturer of HPLC?
Yeah, so this software is for this particular system, right? For the Contichrom system that has two columns and this particular configuration of hardware, pumps, valves. So yeah, the answer is no, basically, because we have here a unique twin column system that can run batch chromatography, right? But other manufacturer systems do not have the twin column capability that's required here.
Okay, and I'm watching our time, so I'll just take one more question. We'll answer the other questions and follow up in writing. So thank you for those. And I think you've touched on this a little bit already, but questioners asked, does this instrument only operate the enrich feature, or does it also perform conventional HPLC functions?
Yeah, so it does run single column chromatography also, right? So it doesn't only do enrich or twin column chromatography. So it has the option to run also other twin column chromatography processes that were mentioned for capture and polishing of proteins, peptides, oligonucleotides. So these have been touched on in the other webinars. So yeah, it's a multi-purpose instrument.
Okay, and on that note, out of respect for everyone's time, we'll wrap up. Thomas, again, thank you for your insights. Very interesting, informative. Thank you to the audience for all your questions, your participation today. If you have any further questions, want to reach out to us, we can be contacted at info@ymcpt.com. That's info@ymcpt.com. And again, in a short while, you'll receive a link to an archived version of this presentation and a link to associated reference documents. Please share these with your colleagues. And as a reminder, this is one in a series of webinars, all of which you can find posted in the resource page of ymcpt.com website, or here on our gracious host, biowebinar.com. We'll end our session now. Thank you and have a great day.
Thank you. Bye-bye.